I don't know the answer to this question. I have some ideas though, which is the purpose of this video. Hello and welcome back to another exciting video with me, Sean, the embryologist. Today we're going to briefly discuss this paper, but it's going to roll more into an open discussion and question section. This video is really in continuation of my previous video where I discussed the study in which they fed male mice a high-fat diet, and then they saw the impacts that had on the epigenetic factors that are added to the sperm, and then what that did to the resulting offspring having issues with metabolism and obesity. So this study is also about the epigenetic factors that are added to sperm after they've been produced. Epigenetics has to do with factors that can regulate gene expression in our DNA. So they can turn on genes, they can turn off genes. It's like a light switch or a dimmer switch. You can turn them on, turn them off, dim it a little bit, brighten it a little bit. And it's a really interesting developing field in regards to genetics and why genes are expressed in the way they are expressed. So first, let's just talk about this paper really quickly. It's a pretty straightforward study. A little bit of background information you have to understand. First, in mice and in humans, sperm first develop in the testes. They start out as these round germ cells, and then they develop into the sperm as we know them, where they have a head and a tail. That's where they complete meiosis, so they're haploid, they only have one copy of DNA. After developing in the testes, they travel to the epididymis. For a long time, we've kind of only thought of the epididymis as the place where they're stored until ejaculation. We knew they finished maturation there, that's where they develop the ability to be motile or to swim. But now we're learning that there's more of these epigenetic factors that are being added to the sperm while in the epididymis. And it's accomplished through these little packages called epididymisomes that come in, they fuse with the sperm, and then they add these epigenetic factors, these quote unquote small non-coding RNAs. Really cool stuff. So this study was pretty straightforward. What they did was they extracted sperm from the epididymis in two locations. One at the beginning of the epididymis, and two at the end of the epididymis. Depending on species, the sperm takes a certain amount of time to travel through the epididymis. And that can range from a couple of days to about 10 to 14 days. I believe in mice, it takes about 7 days for the sperm to travel through the epididymis. Not important. Suffice to say, it takes a certain amount of time to make it all the way through the epididymis. So they have two groups of sperm that they extracted from the beginning of the epididymis and sperm from the end of the epididymis. And then they injected them into mouse eggs. This is, if I didn't make it clear already, this whole study was done in mice. It's a little tricky doing studies in humans, especially around reproduction. And what they found was early development of the embryos was essentially identical. Fertilization was the same, development was identical. As an embryologist, that was my guess. My guess is this is what you're going to see. They're going to develop to the blastocyst stage, which is around day 5, day 6, and they're going to do so at a similar rate. What was very surprising in the findings of this study is that the sperm that were taken from the beginning of the epididymis, those embryos, when put back into mouse uterine, those embryos either A, failed to implant, or B, did not result in live birth of baby mice. Whereas the sperm that were taken from the end of the epididymis resulted in a normal rate of live-born mice. Super interesting. So what they looked at was the RNA profile of the sperm from the beginning and from the end. And what they found was the sperm at the beginning of the epididymis were lacking a lot of these small non-coding RNA epigenetic factors. Conclusion is for mice, sperm that don't have the epigenetic factors added to them at the end of the epididymis fail to have live-born mice, meaning these epigenetic factors are seemingly indispensable for the next generation to be born. And they confirmed the importance of these RNAs because what they did was they injected eggs with sperm from the beginning of the epididymis, made the embryos, and then they injected the RNA profile, the normal RNA profile, into these cells after fertilization, these embryos after fertilization, and then those embryos were able to successfully result in baby mice, showing that, yes, it is in fact these RNA epigenetic factors that are controlling the success of the pregnancies. I made a short video about this study and posted it to my Instagram. And I had other embryologists commented things that were the same thing I was thinking about, which is this. Check it out. In IVF, we can do this thing where we extract sperm from the testes, and then we can use that sperm to inject into eggs. And although at a slightly lower, actually quite a bit lower success rate, we can have healthy live-born babies through IVF. The reason why somebody would need this procedure, there's multiple reasons why. One is obstructive azoospermia. Azoospermia means no sperm in the ejaculate. Obstructive means there's something going wrong with the plumbing. So for example, for many men that have cystic fibrosis, they also will be missing the vas deferens, the tubes that leave the epididymis. So they're making sperm, it just can't get out. So we do this procedure. There's a couple different names for it. Tessie or Tessa. It's just, there's a couple variations on how they get the sperm out, but it's essentially accomplishing the same goal, which is getting some of the tissue from the testes, the seminiferous tubules that are in the testes, taking some of those out, and then hopefully there's sperm in there that we can use and inject into eggs and do IVF with. Side note, if you think this is very damaging for the male testes, it's not. The testes, these seminiferous tubules, it's like miles and miles long of these very small tubules that are tightly packed in there. So when we remove some of them, it's like a drop of water in a bucket. But back to what the embryologists were commenting on my Instagram. How is it that in mice, just taking sperm from the epididymis, not even from the testes, in mice, this fails to result in a pregnancy, but in humans, we can take sperm not just from the beginning of the epididymis, but before they've even made it to the epididymis, and we can make humans with that. That doesn't make sense, especially when you take into account that mice compared to humans are like crazy fertile. So when the embryologist commented that, I was like, this is exactly where my mind went also. How is it that we can take sperm from the testes in humans and have a live birth? But in mice, this requirement is a hard cutoff. I don't know the answer to this question. I have some ideas though, which is the purpose of this video. My ideas, but I also want to get in the comment section and see what your ideas are about why this is. I posted a similar video to this, a much shorter version, to my Instagram, and I opened it up to comments like, what are your guys' ideas and theories? And there were some good ones in there that I hadn't thought of. So for me, something like this, which I think just about everything in biology and nature, you have to look at it through the lens of evolution, through natural selection, through the selective pressures to answer the question, why is it this way? Let me first talk about a gene that is called the histone H4 gene. This gene is present in everything that is living, all animals and plants, you and me. We all have this H4 gene. This H4 gene in our DNA is 306 characters long, and it is almost identical in every single one of us living things. If you compare a pea plant to a cow, there are only 2 characters in that genetic code which are different. The question is why? We think of evolution and random mutations, and that's why there's all these different living organisms, survival of the fittest, etc. Why is it that this one gene has refused to allow any kind of mutation in any animals or plants for hundreds of billions, like Trump, billions and billions of years? This gene says, nope, you are not allowed to change me. I have to stay this way. This may sound a little counterintuitive, but that means there is a higher selective pressure on that gene, meaning variation in that gene, apparently even a tiny amount of variation in that gene, is not conducive to life. Either the plant or the animal can't be born, it especially isn't reproducing. So high selective pressure on either a gene or characteristic equals little to no variation. It must stay the same. Allow me, if you will, to apply this concept to the epigenetic factors being added to the sperm in mice. It appears this is a step that is indispensable in mice. Why? There's another really cool study where they trained mice to fear a specific smell. I believe it was a citronella smell. They trained mice by sadly gently shocking a cage after spritzing this smell. And what they found was the offspring of these mice, and they had all types of controls, so they, it wasn't like the mice somehow communicating to the baby mice. What they found was the offspring of these mice were born with a fear of that smell. Somehow, epigenetic factors were passed down that turned on a gene that said, be afraid of this smell. Citronella is dangerous. So my theory, my thought about these epigenetic factors in mice sperm is that maybe because mice are lower on the food chain, they are much more subjected to predatory behaviors compared to us, the top of the food chain. So them being able to experience their environment, learn from their environment, and then through epigenetic factors, teach their offspring what to be afraid of. They could be teaching them things like eat this type of food, only go out at this time of day. If you hear this sound, run away. This is an indispensable characteristic for the survival of mice. Their biology has said, nope, we cannot shortcut this step. If these sperm do not have small non-coding RNAs, you will not have baby mice. Now let's compare it to humans. Like I said, we can take sperm before they've even gotten to the epididymis, and we can have healthy live-born babies. Why is that acceptable in humans? This is where I want people to get into the comments and present their ideas and their theories. And you can't be wrong because we don't know the answer to that. This is what's kind of fun with this. It's kind of a little thought experiment. And this is why I loved posting this on my Instagram, because people presented some interesting ideas. Like I said, my idea was, okay, well, we're not subjected to predatory factors as much as something that's smaller on the food chain. Maybe you would see this present in many more animals, and maybe the lower you get on the food chain, the more pressure there is on this epigenetic factor in sperm, versus the higher you get, the more predator you are, the more forgiving it is. One concept, one idea that somebody put in my comments on Instagram was maybe because we are able to communicate with words, we're able to communicate more advanced ideas and concepts to our offspring, we can short circuit this epigenetic requirement by teaching them through talk and our advanced brains. To me, very interesting. Like I said, I love looking through stuff like this through the lens of evolution and natural selection. It's impossible to say we know absolutely for certain because we can't go back in time and watch biology evolve, which makes this a little more fun because you can't be wrong. It's a fun guess. I'm excited to read your comments. Tickle my brain, all right? Thanks for watching.